All right then, let's try this one more time. All right, chapter 9, patterns of inheritance. And now we're finally getting to heredity after knowing how DNA works, how mitosis works, how meiosis works. Now we're going to see what happens once we get all these alleles together. It may seem odd that it says your sex was entirely determined by your father, which is the case because the Y chromosome is what designates male in mammals, and since both women and men have an X chromosome, the only thing that's different is that Y chromosome in the male instead of the second X chromosome in the female. So yes, if depending on what you got from dad, you're either male or female. Okay, some features are determined entirely by genes, some by environment, and some by both. Right? So something like the ability to process cholesterol properly is determined by genes. Whether or not you have a certain muscle tone or have a particular speaking voice, right? There's some features that can be changed by the environment, even and also by intention, and some are determined by both. Your chances of inheriting and passing on a genetic disease can often be determined by your family history. So this is why we do the pedigrees like you did in the lab from last time. Now, a great example of what's called artificial selection is domestic dogs, right? People have been selecting and mating dogs with preferred traits for more than 15,000 years. So over those many, many years, that sort of tinkering has led to the really wide variety of body types and behaviors that we see in dogs today. And you think about everything from the tiny Chihuahuas on up to, Great Danes and Grand Pyrenees and all those other giant breeds. Also temperament, right? Some are more nurturing, some are better at tracking. So you know what I mean. Now, the funny part is the biological principles underlying the genetics of that we've been doing all these years in mating dogs, we really have only broken it down recently. Now let's get some vocabulary out of the way. Heredity. Heredity is the transmission of traits from one generation to the next. Genetics is the scientific study of heredity, right? So genetics is actually getting down to what's happening with the genes. Heredity is basically talking about the patterns. Now, Gregor Mendel, who's a monk, worked in the 1860s. And one interesting thing about people who went into monasteries was that you could devote, you basically were stepping away from regular life. And one thing you could do was, in a monastery, some people produced food, baked, brewed, made, generated food. Well, some became scientists. So part of the day was spent in religious activity, and the rest of the day, often people took up some very interesting, in-depth, behaviors. Anyway, so Gregor Mendel, back in the mid-1800s, he found that parents passed on to their offspring discrete genes. Now again, they didn't know what genes were at this time. So he called these things heritable factors. Today we would call them genes, but at the time, again, DNA wasn't known and all that. Now these genes, or heritable factors, are responsible for inherited traits, and genes retain their individual identities generation after generation, which you might tend to think, well, yeah, but previously there were, not a lot of understanding about how heredity worked. And so it was thought that genes could be diluted, right? So the genes could be changed over time, but we now know that's not the case in that respect. Here's Gregor Mendel. This is one of Darwin's contemporaries. Unfortunately, they didn't really get a chance to talk much. All right, in the Abbey Garden. So as I said, Gregor Mendel did his work at a monastery. And in the garden, one thing that proved sort of handy was he tried different plants to work with for his research. Well, he chose garden peas, which have the side benefit of also being, good for food, so not competing for, research with lunch. So why would he choose the peas? Well, A, easy to grow. B, came in many readily distinguishable varieties, right? So things you could compare. Now, a character is a heritable feature that varies among individuals, right? So we would think of things like eye color or height or, ability to process lactose, things like that. A trait is a variation of a character. I guess another way you could look at it is if you think of cars, right? You could have cars have exteriors, right? So the exterior of the car would be the character. The traits would be the different colors you could get that car exterior done in. What turned out to be a wonderful coincidence is that each of the characters Mendel studied occurred in two distinct traits. As you saw from doing the lab and reading this chapter, you realize that not everything comes in two, clear, possibilities. Now, perhaps the most important advantage of pea plants as an experimental model was that he could strictly control their reproduction. Right? If you use things like animals tend to take up a lot of space. You have to be very careful about housing them separately. But using plants, that's a little easier. And plants like a pea plant have kind of an interesting feature. Like many plants, you'll look at this flower here, and you'll see it has structures called stamens and carpels. And the petals are simply, advertising. These reproductive structures, if you notice, this particular flower has both male and female gamete-producing structures. So what you could do is, these are plants that could self-fertilize. In fact, you'll sometimes hear the term selfing, and it's referring to plants that will self-fertilize, that their own pollen would fertilize their own eggs. Well, when Mendel wanted to control things, say he wanted to fertilize one plant with pollen from a different plant, he did it by hand. So instead of having bees pollinating as you would normally see, he did it by hand. You could basically take a very small paintbrush and pick up the dusty pollen from the stamens and then deliver it manually to the carpels. This way, he was always sure of the parentage of the new plants. So, and if you didn't want the plant to self, go to this next slide here. If you didn't want the plant to self, one thing you could do is by removing the stamens from a flower, you ensured that it wouldn't self-fertilize. So again, interesting way to be able to, clearly control fertilization. So in your illustration here, the stamens were snipped off of the purple flower and taken, put out of the way so that those pollen grains would not be involved. He then took stamens from a white flower, picked up the pollen, as you can see in that picture of the brush, picked up the pollen from the stamens of the white flower, and then transferred them over to the carpel of the purple flower. Okay, well, when you fertilize, the eggs in a flower, what you get when that ovary containing the eggs matures is fruit. That's the definition of a fruit. So, looking at the pea fruit here, right, this is now the offspring of those two flowers. So he took the seeds from that fruit, from that pod, and planted the offspring. Now you see a couple of notations used here. One is parents as capital P, right? So you have ways of naming the generations so that you can track, okay, this is the parents, this is crossing those parents, this is crossing those offspring, etc. So parental generation is always referred to as P. The first generation of offspring is referred to as F1, right? Filial one, so the first generation of offspring from that pair of parents. So I'm just going to pop through these. Now, when you cross two purebred varieties, you have what are called hybrids. And again, what is a purebred? You've doubtlessly read this preparing for the labs and all, but a purebred is something where when you mate two individuals, you always get the same offspring. In other words, the alleles, there's no sort of hidden recessive alleles. It's purebred. It has just one, set of alleles. We'll get to this in a little bit. So when you cross two different purebred varieties, you produce hybrids, and that process is referred to as a genetic cross. So you'll hear me saying, when you cross this with that. right? So that's taking two individuals and generating offspring. Now again, the parental plants are the P generation, the hybrid offspring are the F1 generation, and if you cross the F1 generation, now you have the F2 generation, right? So you could see the process here. Now, Mendel performed many experiments in which he tracked the inheritance of characters, such as flower color, that occurs to alternate traits. These results led him to formulate several hypotheses about inheritance. Again, as you now know from doing the labs and your reading, not all inheritance is quite this simple. That's why you'll hear me refer to Mendelian inheritance, which is just plain old dominant, recessive, all that. So these are the 7 characters that he studied. You'll see flower color, flower position, seed color, seed shape. These are all things that have 2 very distinct traits, right? So the characters are the features, the traits are the variations of the seen in that character, right? And you see dominant, recessive, and by now you've read these terms and worked with these terms. So the dominant trait in the flower color would be purple. The recessive trait in the flower color would be white. The dominant trait for flower position would be axial. The recessive would be terminal. And you can follow through the rest of that diagram. So when he's crossing purebred, say purple flowers and plants that are purebred for white flowers, you wind up with, a good way of observing, okay, what are the patterns that are going to take place when you put these 2 possibilities with each other. And this is what they actually look like in the real world. All right, so things he found. So 1, Mendel's law of segregation. And this is described as inheritance of a single character. The next diagram is going to show a cross between a purebred pea plant with purple flowers and a purebred pea plant with white flowers. And he figured out that the heritable factor, what we would call a gene for white flowers, didn't actually disappear in the F1 generation, even though all the F1 individuals will have purple flowers. We'll walk through this in the next couple of slides. So if you see all this offspring have purple flowers, you might think, oh, the heritable characters for the white flowers has somehow disappeared. But he realized that it didn't disappear because when he crossed the F1 plants, the F2 generation, you saw the white flowers again. So he figured out that the gene doesn't disappear in a cross, but it's somehow hidden or masked when the purple flower factor was there. And then he realized, well, the F1 plants must have carried 2 factors for the flower color character, because when you look at the cross of the 2 F1s or F2 generation, you saw white individuals popping up. So from these results and other work, he developed 4 hypotheses, right? So if you see a list, that's something you might expect to see on a test. So again, P generation, these are the purebred parents. F1 generation, so crossing those parents, all flower plants have purple flowers. And when you cross the F1s, you see, if a ratio of 4, 3 out of 4 would have purple flowers, 1 out of 4 would have white flowers. So that's, right there, it was kind of an interesting finding. I actually found a picture of him. I think the original was him holding a flower, but anyway. All right, so Mendel's law of segregation. There are alternative versions of genes that account for variations in inherited characters, right? So if you think of things like flower color, right, you have variations like purple and white. The alternative versions of genes we today call alleles. Again, back then they didn't know DNA or any of this, but the modern term we now use is alleles for the alternative versions of genes. Now, for each inherited character, an organism inherits 2 alleles, 1 from each parent. So an organism that gets the same allele from both parents, therefore having 2 identical alleles for a gene, is said to be homozygous for that gene. So homo, prefix referring to same. Zygous is whenever you see ZYG, that's referring to something to do with that's going to do with a nucleus, right? So an organism that has two different alleles for a gene is said to be heterozygous for that gene, right? So the prefix hetero referring to different. If an organism has two different alleles for a gene, one of those two is going to determine what the phenotype is, what the organism's appearance is. We call that allele the dominant allele. The other allele doesn't have any noticeable effect upon the organism's appearance and is then called the recessive allele. So when you're writing these down, you always want to use uppercase letters for the dominant, lowercase letters for the recessives, and have them be the same letter. So if you were working this out, you wouldn't have a W letter for white and a P letter for purple. You'd have big P for purple, little p for white. Now, a sperm or egg is only going to carry one allele for each inherited character, right? Because the two alleles for a character are going to separate from each other during the production of the gametes, right? Back when you had the reduction division. So we called this the law of segregation, that the gamete is only going to carry one allele for each inherited character. When the sperm and egg unite at fertilization, each is going to contribute the alleles it's carrying, restoring the paired condition in the offspring. And again, most of the time people would say, well, duh. But again, people had different thoughts about how reproduction worked over the years. Back before that time, there was the theory that in the sperm was a tiny, tiny, tiny little person. The fancy word for it is homunculus. And that basically the male delivered this tiny, tiny, little individual to the female, and she was essentially the incubator. So there was a lot of stuff that wasn't quite understood for a while. Anyway, so we're going to look at this diagram showing this. So again, going up to the top, you see the P generation, and again, these are purebred. So if they're purebred, you're going to see two copies, right? So purebred for the dominant, it's going to be big P, big P. Purebred for the recessive, little p, little p. Now, if those individuals are homozygous, they can only contribute one allele, right? It isn't like this or that. It's going to only contribute the dominant allele or the recessive allele. So that means if you have two purebred parents, all that F1 generation are guaranteed heterozygotes, right? They're going to be heterozygous individuals. So you'd say the genotype is heterozygous. The phenotype, what you see, is purple flowers, right? So one thing that will keep coming up are phenotypic ratios and genotypic ratios because they're not necessarily the same. You could have a purple flower that is homozygous dominant, like one of the parents up here, or you can have purple flowers that are heterozygous, like this F1 individual here. Now, when these heterozygous offspring, right, when these hybrids themselves reproduce, if you're crossing them with each other, well, now there's two different possibilities for what's going to be in its gametes, right? So in its eggs or in its pollen grains in the case of flowers. So it's equal chance. 50-50 could either be the dominant or the recessive. So it's like flipping a coin, right? Equal probability, it could be one or the other. Now, we cross those F1s, right? So sperm, which emerge from the pollen grains, when the plant is pollinated, each one of those sperm is going to carry one allele. So it can either be carrying the dominant allele or the recessive allele. Same thing for the female. Each egg is going to have either the dominant allele or the recessive allele. And this thing we have here is called a Punnett square. And again, you've read this by now, preparing for the labs and such. Punnett square is basically a way of mapping out the probable offspring of two parents. Right? So it gives you a way of saying, okay, we know what probability each of these alleles will have. What are the probabilities of certain combinations coming up? So if you look at the F2 generation here, the phenotypic ratio is 3 purple flowers to 1 white flower, right? So phenotypic ratio, 3 purple to 1 white. But the genotypic ratio is not the same. You're going to have 1 homozygous dominant one homozygous recessive and two heterozygous individuals. Right, so the genotypic ratio would be one, and I would use the proper terms: homozygous dominant, two heterozygous, and there's no modifier. Heterozygous just means one of each. So you would never say heterozygous dominant or heterozygous recessive. It's just heterozygous. It's just one of each. And then one homozygous recessive. Right? So if I ask you about phenotypic ratios and genotypic ratios, realize that they're not necessarily going to be the same, because in this case, you can see two different ways you can wind up with purple flowers. So just walking through this. So again, P1 generation, they're purebred, so they're homozygous, in this case homozygous dominant for purple, in this case homozygous recessive, which is going to yield white flowers. And again, because they're homozygous, there's only one possible allele they can make, right? So all from, the gametes from one plant are going to be the dominant, all from the recessive will, the white flowers will be the recessive. So guaranteed, if that's the only possible gametes being produced, then that's the only possible offspring down here in the F1s, right? If both parents are purebred for two different alleles, the offspring are by definition heterozygous. Right, so that's something where you could just read it, on a test, and you wouldn't even have to do a Punnett square. It's like, oh yeah, there's only one possibility here. Now, for that F1 hybrid, it could yield one of two different alleles in its gametes, right? Because now it's carrying one of each. So each gamete is, again, going to have one copy, because you're going to fuse it with the other and go back to the state of two copies. It could either be the dominant allele or the recessive allele. So crossing the F1s, again, here's your two possible, alleles for each gamete in these individuals. And again, it's an equal probability. So, one big P, one little p, one big P, one little p, because it's, again, 50-50 that any one of these gametes will have one or the other. So the possible combinations are, if one parent contributes the dominant allele, the other contributes the dominant allele, now you have a homozygous dominant individual. The phenotype is going to be purple flowers. Well, if you cross that dominant allele with this recessive allele, you still have purple flowers, but now the genotype is that of a heterozygote. Okay, same thing here, second heterozygote. And this last one, if you cross the two recessive alleles, if you have two copies of the recessive allele in this case, now you see the recessive trait, right? In this case, the white flowers. So again, don't forget phenotypic ratio versus genotypic ratio. And as we've been using already in this chapter, Punnett squares are going to highlight the four possible combinations of gametes and the four possible offspring in the F2 generation. So each one of those four squares represents an equally probable product of fertilization. And again, we distinguish the products of these matings as what the phenotype of the offspring are and what the genotypes of the offspring are. So Mendel found that each of the 7 characters he studied had the same inheritance pattern. So every time he had something with there were a character with two different traits, whenever he did these patterns of crossing purebreds and crossing the hybrids, he kept seeing the same pattern. So it wasn't something that just happened in flower color or just happened in pea pod texture. So he found each of those 7 characters had the same pattern. One of two parental traits disappeared in the F1 generation, only to reappear in 1/4 of the F2 offspring. So the mechanism behind this is explained by Mendel's law of segregation. During gamete formation, pairs of alleles segregate, and the fusion of gametes at fertilization creates allele pairs again. So remember that whole business of independent assortment, that during meiosis, when these guys are lining up, you may have all, of one parent on one side and all. We'll get to this in a second. We'll return to this in a moment. Okay, so back to law of segregation. So we talk about homologous chromosomes, and you remember this back from mitosis and meiosis, right? You had these paired chromosomes, one contributed from each parent. And alleles, as you know, are the variations of a character. So the diagram we're going to see on the next slide is going to show a pair of homologous chromosomes, so chromosomes that carry alleles from the same genes. The location of a gene on a chromosome is a locus, right? So locus is the location. The plural is loci, L-O-C-I. Now, alleles, the alternate versions of a gene, reside at the same locus on homologous chromosomes. So they're at the same physical address, if you will, on each of those strands. That again, the two chromosomes may bear either identical alleles or different ones, depending on whether it's homozygous dominant, homozygous recessive, or heterozygous. So we look at this guy here. So this is a pair, right? So this is the contribution from two different parents. So these are homologous chromosomes. The same genes are contained along the length of each of these chromosomes. Now we're going to look at three loci. So at this locus, the individual is the offspring being produced is going to have two copies of the homozygous of the dominant allele, right? This is homozygous dominant, right? So if they got two of the same allele and it's dominant, that's homozygous dominant. At this next locus, you see two recessive alleles. So if each parent contributes the same recessive allele, then you have homozygous recessive. And then the last possibility, of course, is one contributes the dominant allele, one contributes the recessive allele, and this would be heterozygous, which again is defined by one dominant and one recessive allele. So Mendel figured this out. He deduced this law of segregation by following one character through two generations. So when you cross two individuals that are heterozygous for one character, we call that a monohybrid cross, right? So you think of that F1 generation of those flowers, right? They're each heterozygous. They're carrying one allele for the dominant, one allele for the recessive, right? So if you cross two individuals with that makeup, they're heterozygous for that one character, we call it a monohybrid cross. Now, the question would be, well, if that's what we see in a monohybrid cross in the Punnett squares we've looked at so far, what would you get from a dihybrid cross, right? A cross between two organisms that are each heterozygous for two characters being followed. All right, so again, the checkpoint. If they have the same genotype, must they have the same phenotype? No. Right? You could have, if two plants have the same genotype, must they have the same phenotype? If the genotype is the same, then you would typically see the matching. So in other words, you flip that question around. If they have the same phenotype, must they have the same genotype? And that would be no, right? You could have a purple flower that's either homozygous dominant or heterozygous, right? So the genotype determines the phenotype. So if they have the same genotype, they have the same phenotype. But you could have different genotypes resulting in the same phenotype. So this question was answered when Mendel crossed the F1 plants with each other, right? The first generation. If the genes for the two characters were inherited together, then the F1 hybrids would produce only the same two kinds of gametes they received from their parents, and the F2 generation would show a 3-to-1 phenotypic ratio. If, however, the two seed characters sorted independently, then the F1 generation would produce 4 gamete genotypes in equal quantities, and the F2 would have 9 genotypes. Now again, we're talking about a dihybrid cross. So the monohybrid cross was what we've seen before. Now we're trying to think, what would we see in a dihybrid cross? So if they traveled together, you would see that same ratio, right, that you saw in the original monohybrid cross, right? One phenotype, one genotype, and then the crossing those, you had that 3-to-1 ratio. But would this happen in a dihybrid cross? If they sorted independently, then you wouldn't see that 3-to-1 ratio. The F1 generation would produce 4 gamete genotypes in equal quantities, and the F2 generation would have 9 genotypes, producing 4 different phenotypes in a ratio of 9:3:3:1. Now, I'm going to tell you right now, I will not make you have to sketch out a dihybrid cross. Do, however, remember this ratio of 9:3:3:1, because that is distinct to this type of cross. So the Punnett square we're going to see in the next slide is going to show that a dihybrid cross is basically, in terms of statistics, the equivalent to two monohybrid crosses occurring at the same time, right? Because everything has its own separate probability. So if we look here, now one hypothesis would be dependent assortment, right? That in the parent generation, you'd say, okay, we got homozygous dominant for both of these characters, homozygous recessive for both of these characters. So again, okay, this is so far like what we saw in the monohybrid cross. The only possible gametes that are contributed from the parents are going to be, the same. So here's your F1. So it's heterozygous for both characters, big R, little r, big Y, little y. If the alleles had to travel together, if dominant could only travel with dominant, recessive only travel with recessive, this is what you'd see, right? You're back to that 3-to-1 ratio. But as you can see from the international no sign, that's not what you see. What you actually see is this. All right, so again, purebred parents, heterozygote hybrid, F1 generation, but these are the possible gametes and crosses that you get from those two, from crossing two of those individuals. All right, so 9 out of 16 would have both dominant traits, right? So round seeds, yellow seeds. 3 out of 16 and 3 out of 16 would be the two possible heterozygotes, right? Dominant shape, recessive coloration, recessive texture, and the dominant color, right? So 9 with the phenotype of round yellow, 3 each for the two possible heterozygotes, and then 1 out of 16 would have gotten recessive alleles for both of the characters. So you can see if you're trying to breed for something like this, this parent we have up here, it's going to take quite a bit of breeding to be able to get individuals to cross to generate that purebred. All right, so again, the two hypotheses would be dependent assortment, that the dominant alleles travel together and the recessive alleles travel together, or independent assortment, that these alleles will move separately. So in other words, when you generate the gametes, you don't see this. You don't see the dominants travel together, the recessives travel together. What you actually see is this. All right, so now you can see the 4 possibilities, right? This is going to be homozygous, and this is going to be dominant for both of the alleles, right? Big R, big Y. This is going to be a heterozygote for, in terms of, I'm sorry, I'm not saying this right. You're going to have one that's carrying both dominant alleles, one where you have one dominant and one recessive allele. Here's the other possible one dominant, one recessive allele. And again, that last possibility is 2 copies of the recessive allele. Now, because of dominance, you're going to have anything where you have a copy of the big R, it's going to be round, right? So now you can see all the possible crosses where you have the dominant allele showing up. Well, anytime you've got the dominant allele, right, for the, in this case, the shape, right, if you have a big R, little r, or a big R, either way, the phenotype's going to be round. So that's why I was talking about knowing that 9-3-3-1, because this is the ratio, the ratio that you see in this type of dihybrid cross. 9, both dominant, 3 each for one dominant, one recessive, and 1 out of 16 carrying both recessives. And this is what they actually look like. These are things you actually do in biology labs, is you get these P's and you count them up and work out the ratios. Obviously, we're not doing that here, but just wanted you to see what they actually look like in real life. All right, so Mendel's dihybrid cross supported the hypothesis that each pair of alleles, right, you're tracking 2 different characters, each pair of alleles segregates independently of the other pairs during gamete formation, right? So the inheritance of one character does not control or have an effect on the inheritance of another, right? So you can't say, oh, if it has round, round texture, it's going to have yellow color, right? It could be round texture and yellow or green, right? So each is inherited independently. And this is what we call the law of independent assortment, that alleles travel, segregate independently. And they give a great example of independent assortment in the inheritance of two characters in Labrador retrievers. Right, so here's our labs. Let me go to the next one just to work out these guys. So you have, let's go back to the original, sorry. So the phenotypes, black coat is dominant, right? So black coat and normal vision. Normal vision is dominant. And believe it or not, there are genetic forms of blindness. So in these dogs, we see this. So phenotype is black coat, normal vision. So you know there's one copy of the dominant allele. Black coat and blind means, okay, you've got one copy of the dominant allele, right? Because that character is, that trait is dominant. But if the dog has this type of blindness, then it has to have two copies of the recessive allele. Moving on to this guy, recessive color, right? Chocolate coat, so two copies of the recessive allele, but normal vision, which means you have at least one copy of the dominant allele. And then the double recessive here, you've got chocolate coat and blind, right? So if we actually bred these dogs, you would see this pattern emerge where you'd have out of 16 offspring. Now again, obviously the number of puppies varies. You'd have several matings and count up puppies over a number of dogs, but just if we magically had 16 in the exact predicted ratio, 9 black coat with normal vision, right? Both dominant both dominant traits. 3 black coat blind, right? So this is one of the heterozygous combinations with the dominant, in this case for the coat, the recessive for the vision. The other heterozygotes are going to have the recessive color, the chocolate coat, and the dominant character of the dominant trait of the normal vision. And again, 1 out of 16 is going to have that, combination of both recessive alleles. Now, you might find yourself asking, okay, when we have a dominant phenotype here, right, the black coat, we only can guarantee that one of those alleles will be the dominant. Whereas if you have something with the recessive trait, right, so chocolate coat, you know it's 2 copies of the recessive allele. There's no other way to get the chocolate coat. But here, you could have 2 possibilities, homozygous dominant or heterozygous, right? Either one of those would give you, the dominant phenotype. Same thing for the vision. You could have normal vision with just 1 copy of the dominant allele. You could have, and the second be the recessive allele. You could have another with 2 copies of the dominant allele, right? So when you have a dominant phenotype, right off the bat, you don't know the whole genotype. You only know that it has 1 copy each of the dominant. So passing through this crowd here. Now, so you want to ask yourself, sorry, let me go back. Well, how do you figure this out? How do you know what the second allele is in an animal that's showing the dominant trait? Well, what you do is you say, okay, we're going to cross this individual with the dominant phenotype but unknown genotype with a homozygous recessive individual. Because if you have a homozygous recessive individual like that dog here, you know its exact genotype, right? If it's carrying both recessives, it's 2 copies of the recessive for each of those characters. So you know exactly what this guy has, so you can use crossing this with these others to sort out what the second allele is. We call this a test cross. It's a mating between an individual dominant phenotype but unknown genotype and a homozygous recessive individual, because you know exactly what alleles that guy is going to produce. So this next figure shows the offspring that could be the product of such a mating. Right? So here's our and we're just looking at color, the coat color at this point. So we have a chocolate colored dog. We know that's 2 copies of the recessive allele. Here we have a black dog, and all we know is there's at least 1 copy of the dominant allele. All right, so you cross these two, and what are your 2 possibilities? If that second allele is dominant, right, because this other guy, the chocolate guy, is only going to contribute the recessive. So if the second one is going to be a copy of the dominant, then what? It's hard to point and not tap, sorry. You have, if you have 2 homozygous parents, one homozygous dominant, one homozygous recessive, then what? They can only contribute the dominant or the recessive allele, and all the offspring you'd see from crossing those 2 dogs would be black. If the second allele was recessive, when you cross that recessive individual, that homozygous recessive individual with what would be a heterozygous black dog, then you'd see black and chocolate show up. So the only way you'd see chocolate offspring would be if that second allele was recessive. If you only saw dominant, then what? This guy was contributing to the recessive, so it has to only be contributing copies of the dominant. So it's kind of neat. It's a way of testing. So people who are breeding animals, it's useful to figure out just what genotypes you have in the offspring. Now, Mendel was fortunate in that he also had a background in math. And so this helped him understand patterns of inheritance, right? He understood that genetic crosses obey the rules of probability, that the same rules apply when tossing coins, rolling dice, drawing cards, right? These same rules. The rule, and this is an awkward sentence, the rule of multiplication states that the probability of a compound event is the product of the separate probabilities of the independent events, which is very hard to read. Let's see this in a picture. So here's our F1 genotypes, right? So both are heterozygous. So when they form eggs and sperm, it's 1 out of 2 chance it will be either the dominant or the recessive allele, right? Like a coin toss, it can either be heads or tails. All right, same thing for the male here, right? The gamete can either be bearing a copy of the dominant or a copy of the recessive. Looking at the possible crosses, all right, if this is 1/2 probability that you get this and 1/2 probability you get this, okay, 1/2 times 1/2 is 1/4, right? So 1 times 1 is 1, 2 times 2 is 4. So the probability of getting 2 copies of the dominant is 1 out of 4. Similarly, 50% chance you'll get a recessive here, 50% you get a recessive here. So another 1/2 times 1/2, 1/4 probability of getting that homozygous recessive. Now here we have the calculation, same calculation, right? 1 out of 4, it's the same product each time. But because these are both, slightly different, okay, you're going to have 2 chances out of 4, right? Or 1/2 the chances of getting a heterozygous individual. Now, you normally would never write the recessive allele first, but because of the business with drawing this out with coins, one of them was going to be written sort of wrong. You typically put the dominant allele first. So that's written a little backwards, but you get the same result. All right, so if you're studying people, obviously you cannot control the mating of their research participants. That's frowned on. So instead, they've got to analyze the results of matings that have already happened. Right? So when you worked out those pedigree examples, you can do is you can see how, as far back as you have information for, you can observe what parents, grandparents, etc. had. So a geneticist tries to get as much information as possible about a family's history for a trait. Then the researcher can take that information that, oh yeah, great-grandma had this and uncles, had that, assemble that information into a family tree called a pedigree. And because of Mendel's laws, this business of probability and independent assortment and all, we can deduce the genotypes for most of the people in the pedigree. So going to this example. So this is a pedigree showing inheritance of freckles versus no freckles. right? So freckles are dominant, so the genotype could either be homozygous dominant or heterozygous, right? So this is the first generation in our genotype up here. If a person has no freckles, then it's carrying 2 copies of the recessive. So here we have the first generation, in this case grandparents, second generation parents, aunts, and uncles, and third generation, in this case, is only 2 individuals and their siblings, their sisters. Now, if you look at the key here, so female is designated by round, male is designated by square. The dominant is not filled in, the recessive is filled in, because if you think about it, you're less likely to see the recessive, so you want that to stick out. So going up here, we have 2 parents that are heterozygous. You might say, well, how do you know that? Well, because some of the kids do have the recessive trait. Right? The only way you can get 2 copies of the recessive is that each parent has contributed one. Right? So if you see an individual with the recessive trait, then okay, the parents have to be hets. Right? They have to both be contributing a copy of that recessive allele, even if their phenotype is that of the dominant. So this individual showing the dominant trait, right, having freckles, in this case a female, it's round, that person's phenotype can either be homozygous dominant or heterozygous. Same thing here. There's a son that is showing the dominant trait, so he has to be, if we know, I might say, if you originally were drawing this out, you'd have this FF or FF. The reason why he's known is because we're going to look back on his partner's tree here. So going back to the grandparent generation here, okay, we have the father with no freckles and the mother with freckles. And looking at the offspring, right? So what are the probabilities that you're going to see some with freckles and some without? So looking at the offspring, now this individual has freckles. You do not have to see offspring, right? If this person has freckles, they have to have 2 copies of the recessive allele. So even though Julia did not have offspring herself, you know her genotype, because she has the recessive trait, had to be homozygous recessive. So if you have a homozygous recessive offspring, then each parent has to have at least 1 copy of the recessive allele. If you see the recessive phenotype, then it's 2 copies of the recessive. If it's the dominant phenotype, then it's a heterozygote. So now we look at the other offspring, Ina, and we have 2 individuals now showing freckles, right? They both have the dominant phenotype. But until we look at their children, we don't know if they're heterozygous or homozygous dominant. So we look down here and 2 children, and fortunately, one is showing the recessive, no freckles. So if you have a kid with no freckles, at least 1 copy of the recessive allele had to come from each parent. So we now know, even though their phenotype is dominant, their genotype is heterozygous because they have a kid that manifests the recessive. Right? So we look at the other offspring, and now this would be a case where if you did not have this person cross with anybody else, you would not know, just looking at them, if they were homozygous dominant or heterozygous, right? It's only when there are offspring that you can hopefully see the recessive turn up, and that would tell you, right? So it's kind of a nice way of, if you can get enough history of individuals to be able to sort out, what their genotypes are. And in some cases, if there's something like a disorder that's in the family, it helps to be able to do this, to be able to see the possible combinations that you would get from the parents to see if there's a probability of something being passed on to the offspring. Now, a trait that's dominant does not imply that it's either normal or more common or good, right? So it doesn't really give any weight to, that trait versus the recessive. It's just that dominance means that a heterozygous genotype results in the dominant phenotype. Now, sometimes you hear the term wild type traits. That's what you see most often in nature, right? So this is what's happening out in the real world, not being bred in a garden or something. So wild type traits are not necessarily specified by dominant alleles. We'll get back to this in a sec. Now, the genetic basis of many human characters, like eye and hair color, aren't really understood as well because they're complicated. You don't have two options, one clearly dominant, one clearly recessive. So Mendel's laws can be applied to the inheritance of human traits that are generally agreed to be determined by a single gene, like freckles or the widow's peak, pointed hairline. Right? We generally accept that there are two possibilities, and it's only one gene that determines this. Now, when we get to the human genetic disorders in the table in a couple of slides, you're going to see them inherited as dominant or recessive traits. Now, these disorders therefore show simple inheritance patterns like the one Mendel studied, right? So one gene is going to have two possibilities. And just another example of inherited human trait thought to be controlled by just one gene, right? So dominant is the widow's peak, recessive is the straight hairline. Now, these are autosomal disorders. What was autosomal again? Autosomal referred to genes that were not, chromosomes that were not the sex chromosomes, right? So everything but the sex chromosomes were the autosomes. And we see two groups here, recessive disorders and dominant disorders. So something like albinism, you'd have to have two copies of the recessive allele for pigment to show, for this type of pigmentation, I should say. Not all pigment is going to be one gene. But the thing that's determining melanin deposition, if the person has no pigment in the skin, hair, and eyes, that person would be an albino. And the only way you get that is from two copies of the recessive allele being carried by each of the two parents. Cystic fibrosis, it is a dreadful thing. It generates excess mucus in the lungs, in the digestive tract, the liver. It's hard for the person to expel the mucus, and because you're not clearing out the mucus, there's increased susceptibility to infection. It's often lethal in early childhood unless it's treated. PKU, this is someone who can't process phenylalanine. And because they can't process phenylalanine properly, it's an amino acid that you find in food, then it builds up in the blood. And that leads to problems. As far as what you'd see in phenotype, there wouldn't be normal skin pigment, and unless it's treated, you will see developmental disabilities. Kind of a fun fact, if you buy food, in this country that has NutraSweet in it, one of the components of NutraSweet is phenylalanine. And you'll see on the label, warning, PKU, this contains phenylalanine. Because if you do have phenylketonuria, you have to avoid consuming phenylalanine, right? Because your body's not going to process it. But in Canada, they don't have that legal element. They have it. Their element is that not regarding NutraSweet, but regarding anything containing saccharin. So it's kind of interesting. Depending on where you travel, if you do have a digestive, or some sort of nutritional challenge, you have to be careful because not all countries label the same way. Okay, onward. So sickle cell disease, this one from previous chapters, right? So this is a disorder where you had a substitution, and the result being, misshapen red cells and damage to tissues. Last one here is Tay-Sachs disease. This is another one where you see lipid accumulation in brain cells, and so unfortunately it is typically lethal in childhood. But these are all autosomal disorders that are manifested if an individual gets two copies of the recessive allele. So you might ask, well, what the heck is going on here? Dominant disorders. Well, we have here 4 dominant disorders being described, one being achondroplasia. Chondro refers to cartilage. It's a problem with the cartilage of the long bones of the body. It's a type of dwarfism that's inherited. Typically, the individual will have short, if you think of the long bones, these are the limb bones, not the axial skeleton of the head and spinal column, etc. So one of these individuals will tend to have a basically normal head and torso, but the arms and legs will be short. So this is one type of dwarfism that's inherited. One type of Alzheimer's disease is a dominant disorder. Huntington's disease is kind of a dreadful thing. It's the individual has uncontrollable movements and cognitive impairments, and it really doesn't kick in until middle age. So you could go through your whole youth not knowing, and then you'd see the symptoms popping up later in life. And another one here is hypercholesterolemia, and as you can guess, hypercholesterol, right? So this is excess cholesterol in blood because the body can't manage it properly. So it's not because you're eating too much, it's because your body can't pull cholesterol out of the bloodstream. We see this associated with heart disease. Now you might say, well, Huntington's, this is terrible. This is, a horrible disease. Why is this allele still around? Think about when it starts taking effect, middle age. By this point, the individual, if they didn't know their genotype, would already have had children. And by the time they reached middle age, if they were going to have children, they had already had them. And so that's why the allele doesn't just vanish because it's such a terrible disorder to have. It just kicks in at a point where you don't know. Same sort of thing here with this type of Alzheimer's. Usually strikes late in life, so the person's already had children. So yeah, it's a dominant disorder, and if their children, it's very probable they passed it along to their children. Dwarfism is kind of interesting. This type of dwarfism is interesting because 2 copies of the dominant allele, homozygous dominant, is lethal. The individual does not survive, is never born, or it never completes development. So if you see an individual with achondroplasic dwarfism, they have to be a heterozygote because the dominant is going to determine having that particular trait. But if they're alive, it meant that the other was the recessive allele. So kind of interesting stuff when you start looking at dominant disorders. But most genetic mutations that cause medical disorders are recessive, right? So individuals who have the recessive allele but appear normal, right? So this would be a recessive disorder. If they have the recessive allele but appear normal, they're carriers, right? They could potentially have offspring with the disorder if the person they have children with also is also a carrier, right? Is also carrying a copy of the recessive, even but they appear normal. And cystic fibrosis is it is the most common lethal genetic disease in the United States. About 1 in 31 Americans, or about 10.5 million Americans, are carriers. So the allele is out there. So this is one where it would be important to know family history to know if this character was there. Now, using Mendel's laws, right, straight Mendelian, normal, 2 possibilities, 1 gene controlling a character, we can predict the fraction of affected offspring that is likely to occur from a marriage between 2 carriers. So let's say we're looking at this individual one. Predicted offspring with both parents are carriers for albinism, right? So normal pigment in both parents, but they are carriers. So it doesn't affect their phenotype, but they are carrying that recessive allele. So what are the possible gametes, right? Big A, little a, because they're both heterozygotes. So normal vision in terms of 2 copies of the dominant allele, normal vision from being heterozygous, and albino because they both got 2 copies of the recessive allele. Now they could have had, statistically, right, it's 1 out of 4 you get this, 1 out of 4 you get this, 2 out of 4 you get this. So the odds are 3 out of 4 that you'll have a child with normal skin pigment. So again, keep in mind, these are all equal probabilities. You can toss the coin, or you can perform the cross here, and the first child is albino, right? It's not something where you have to have 4 kids and one of those 4 is going to show albinism. They could have 6 kids and all of them have normal pigment. when you hear of something skipping a generation, it's typically where you see something like these parents, you see no albinism in the parental generation, but it pops up in the F1 generation or the offspring of those parents. Sometimes you could have something that's carried around for a while and you don't see the recessive pop up. Um, a person I know had red hair, which was recessive in that population of people, and he had an albino aunt, and so it's kind of interesting. It's like I always kind of wanted to be able to do a pedigree, but never got a chance to do so. Anywho, so if you have a couple of individuals who are both carriers, and in this case they had 3 children without cystic fibrosis, if they have a fourth child, again, each reproductive event is a new probability event. So the probability would be what, 1 out of 4, or 25%. You could look at in terms of percentage that the child will have that disorder. Now again, some disorders are caused by dominant alleles. So achondroplasia is again one form of dwarfism. Well, the homozygous dominant genotype causes death of the embryo. So only heterozygotes will have the disorder. They can only have one copy of the defective allele. So that means a person with achondroplasia has a 50% chance of passing the condition on to any children. Let's take a look. All right, so here's the Punnett square. Normal, no achondroplasia, is going to be what? 2 copies of the recessive. This is a dominant disorder. The other parent having achondroplasic dwarfism is, by definition, a heterozygote. So now, it's like, okay, one parent is homozygous recessive, one parent is heterozygous. The possible offspring are, here's a heterozygote showing dwarfism, here's another heterozygote with the dwarfism, one individual that's normal, and another individual that's normal. So in this case, you have basically a 50-50 chance that any one child will be either normal or manifest that type of dwarfism. And I remember there was a show on TV where it was basically following a family, and one parent had achondroplasic dwarfism. I think it was the mother. And it was kind of interesting because I guess the show was around long enough. They produced children along the way and had a chance to see that you can see both normal height children and children with achondroplasic dwarfism. Now, the dominant alleles that cause lethal disorders, right? Achondroplasia is not a lethal disorder. Dominant alleles that cause the lethal disorders are much less common than a lethal recessive, right? Because one copy and you've got a problem. So again, back to Huntington's. This is a degenerative disorder that doesn't start till middle age. So if you didn't know your genotype, if you didn't know your family history, say you were adopted, and you would know, information about the family that child came from, well, then that individual would go through life and not know until middle age. And that's when the degeneration begins. So, and again, this is something, it can't be reversed, it can't be fixed. It's a terrible disorder to have. Now, because the allele is dominant, any child born to a parent with the allele has a 50% chance of inheriting the allele and with it the disorder. So Huntington's is a situation where, if you think about it, this is a terrible choice because now you can be tested. You can find out. If you have Huntington's in your family, you can actually find out if your genotype is going to be containing at least one copy of that dominant allele. But the question would be, would you want to know? If you intend to have children, I would say definitely, because then you would have to think about, would you want to risk having children who are that, had that, but your parents had you, right? And so an individual has this strange choice in the modern era where do you not find out and just live your life, or do you find out and know, one way or the other. Now, again, anybody could get hit by a bus tomorrow, but it's just the fact that that people with these disorders like these, it used to be something you just worried about, but now you can know. But then the question is, do you want to know? Anyway, that's a whole nother course. So, short legs and dogs, right? For some reason, we like to have dogs with short, little tiny legs. So, domesticated dogs show a ridiculous range of phenotypes, right? And one thing that you see is chondrodysplasia in dogs, and that results in shortened bones, right? So it's another thing that affects the length of the long bones. So genetic researchers are like, okay, let's see how this works. So the researchers identified a specific change on chromosome 18 in dogs that corresponded to short legs. And we're able to link that gene in dogs to a related protein that's associated with the most common type of human dwarfism, right? Because we're all mammals. There are things that are shared. In this case, that particular gene, and the problem of short limbs. Now, looking at dogs, all right, we got some guys here with short legs: the Corgi, the Dachshund, the Basset Hound, and we got some breeds with long legs, right? German Shepherd, Greyhound, Beagle. So they compared the DNA, right? So they had 95 dogs from 7 short-legged breeds, 702 dogs from 64 long-legged breeds, extract the DNA, examine the chromosomes, and compare, right? Is there a pattern you see in these that is different from the pattern you see in these? So popping over the pictures here. Oop, I lost a picture. Oh, sorry. All right. Anyway, I thought I had a bigger picture that pointed out the detail, but anyway, but it turns out that, okay, there is a genetic basis to that type of limb. All right, so genetic testing. Now, as pretty much all of you know, there are many ways to identify genetic diseases. There are about 30 of treatable genetic diseases that can be identified before the disorder appears. And you have things like technologies to detect genetic disorders during pregnancy. Usually, genetic testing before birth requires getting some fetal cells. Once those cells are obtained, they can be screened for genetic diseases, right? Where if things like, you think of trisomy 21, right? You could see a karyotype from that individual and see, oh yeah, there's 3 copies at 21. So some things can be determined from fetal cells. One way you can get cells is amniocentesis, taking a needle to extract the fluid that surrounds the fetus, because cells are shedding off into that, and you can test that. Another type is chorionic villus sampling, where the physician inserts a little tube through the vagina, through the cervix, and then removes some placental tissue. They have a way of detecting, tissue there. Or a new one, which is not invasive, is involving isolating tiny amounts of fetal cells or DNA released into the mother's bloodstream. Right? All these previous technologies involved invasive things that could be disturbing to the pregnancy. Right? Now we're developing things where you can get it from the blood. They're very accurate. You can form them much earlier, and they're much safer. So they're gradually replacing those other screening methods. And again, depending on what character you're talking about, you have choices, right? But that can be a difficult choice. All right, variations on Mendel's laws, because life is never that tidy. So Mendel's 2 laws explain inheritance in terms of genes that are passed along from generation to generation according to nice, tidy rules of probability, right? 50-50 chance and all that. And those laws are great for all sexually reproducing organisms, so plants, animals, fungi, etc. But Mendel's laws don't explain everything. In fact, for most sexually reproducing organisms, cases in which Mendel's rules can strictly account for the patterns of inheritance are kind of rare. And that's what I meant about the pea plants being sort of a fortunate discovery, because if you had started with organisms with very, more complicated genetic patterns, he might not have worked out the things he did. So more often, the observed inherited patterns of of characters are more complicated. Now, if I talk about simple Mendelian genetics, I'll talk about what's called complete dominance, right? That's the straight up two possibilities, one dominant, one recessive. But here we have something called incomplete dominance, where the F1 hybrids have an appearance in between the parental phenotypes. So hypercholesterolemia, as I mentioned earlier, it's a human trait that is an example of incomplete dominance and is characterized by dangerously high levels of cholesterol in the blood. So you have a person that is homozygous for the dominant character, that's normal. A person who is homozygous for the recessive is, and this is a disorder where you don't have receptors on the cells where normally cholesterol is being pulled out of the blood. So you have a normal number of receptors, you're normally pulling cholesterol out of the blood. If you have the homozygote, the homozygous recessive, then they have about 5 times the normal blood cholesterol because they don't have those cell receptors. So they're not pulling cholesterol out of the blood, and it builds up, right, and gets associated with things like heart attacks at a very early age. But the heterozygote, it's not just showing, if you'd say, oh, if it's regular dominance, they'd have normal, but they don't. They don't have the normal number of receptors. They don't have, 0 receptors, but they're about in between. So the blood cholesterol levels are about twice normal. They're literally, incomplete dominance. You're seeing a phenotype in between the two parental types. Let's see this in flowers, right? So we have a red and a white flower, and these are purebreds. The F1 generation is pink, and you're like, wait, what? It's not red, it's not white, it's pink. That's because it's not complete dominance. You don't see red flowers. You see something in between the two parental phenotypes, right? So you have red flowers, white flowers, and then the first generation is pink, right? So the heterozygotes are going to be in between the two homozygous conditions. Now, if you cross these guys to each other, it gets more interesting than the Punnett squares we'd seen before. Let me pop ahead to the bigger illustration. Not much bigger. All right. So if each of these individuals is heterozygous, homozygous dominant will appear red. Homozygous recessive will appear white. The heterozygotes are going to be pink. So instead of that 3-to-1 ratio we saw with simple dominance, you see this 1-to-1 ratio in the phenotype. Oh, I do have an illustration of this. Good. Yeah, this is the hypercholesterolemia. I know, I stumble on the small words, I don't stumble on the big ones. So genotype, homozygous dominant, they can make the LDL receptors, and that pulls the low-density lipids out of the bloodstream. Homozygous recessive, they don't have the receptors, right? So the cholesterol builds up very quickly. The heterozygote is not showing the dominant, it's not showing the recessive, it's in between. So it's a mild form of the disease. Now the next thing is going to be something called codominance, right? So incomplete means the offspring is, the heterozygote is in between what you'd see in the two homozygous states. ABO blood typing, you actually have 3 alleles of a single gene. So now you're thinking, okay, this makes things a little more complicated. Various combinations of those 3 alleles produce 4 phenotypes. And as most of you probably know already, a person's blood type may be, and this is without regard to Rh, right? We're just looking at the ABO. A person's blood type can be A, B, AB, or O. And what those letters refer to are carbohydrates called, they designate A and B, that are found on the surface of red blood cells, right? So these are carbohydrates that your immune system, recognizes. Now, because there's 3 alleles, there are 6 possible genotypes, as we're going to see in the next illustration. So, both the I superscript A and the I superscript B alleles are dominant to the little I allele, right? So big I is dominant, little I is recessive. Big I comes in 2 flavors. Big I superscript A and big I superscript B. So a person who has 2 copies of A, or 1 copy of A and 1 copy of I, right, so heterozygous, have type A blood because you see that dominant. Same sort of thing here, where you have 2 copies of the dominant in the case of I superscript B, and then as I superscript B and a person bearing the little I, they have type B, right? So regular dominance in that respect. If you have 2 copies of the recessive, so recessive homozygotes have type O blood because O, like 0, neither carbohydrate marker on the surface. Now, the last one gets a little funkier because now we have, what, 2 types of dominant alleles. And the people who have this genotype of superscript A, big I superscript A, big I superscript B, they make both carbohydrates. So it's not one or the other or something in between, it's both. And that means that big A and big B alleles are codominant. So both alleles get expressed in the homozygous individuals who have type AB blood. So let's go to the illustration. So again, our blood groups, these are the phenotypes, right? A, B, and O. So because A and B are both dominant alleles, you could have either homozygous for the A allele or heterozygous, right? With little I, the recessive, right? So either way, you manifest the dominant allele. Same thing here. Because B is also dominant, you could have homozygous or heterozygotes, and you're still going to see the B phenotype. AB, this is what tells you it's codominant. It's not, this carbohydrate or that carbohydrate or something in between, it's both. So in this case, the heterozygote that has both the dominant alleles is going to be showing both of those carbohydrates. Now, with regard to donation, right, everyone thinks of O as the universal donor. Why is that? Well, individuals, depending on the carbohydrates on the red cells, will have things called antibodies in their blood that are going to be complementary to this. For example, if a person has carbohydrate A on the red cells, they're going to have anti-B in their blood. So that way, if cells came into the bloodstream that were B blood, the antibodies would clump them up. They'd bind them, and you wind up forming clumps and clots, right? So there would be an immune response against that. So a person who has A has anti-B. Conversely, if a person has B, they have anti-A. A person who has AB has neither, right? Because they get both carbohydrates, so they don't have either antibody. So if you separate blood, so you have the packed red cells and then the fluid called plasma, AB plasma is the universal, right? Anybody can get AB plasma because there's neither of the antibodies in the blood. Now, a person can receive, pretty much everybody can receive O cells because there are neither of the 2 carbohydrates there, right? So if you have no carbohydrates, there's nothing you're not going to have something for the recognition on those cells. But you don't have A, you don't have B, but you have both of the antibodies. And this is why anybody can receive O blood, but a person who is O can only receive O blood, right? So this is compatible with any of these guys, but they can only receive the same type because anybody else's red cells, rather, are going to be a problem. Looking at this a slightly different way. This used to be something routinely done in classrooms. Obviously, in the day of blood-borne diseases, this all went out of fashion. But I think they have blood simulators now, so you can do it. But not the old school where you pricked your finger and tested yourself. Anywho, so here's our phenotypes. So if you take blood from each of these groups and mix it with antibodies from this group, you get the following reactions. Now, if it's A, what's going to be the antibody? Anti-B. So a person who's O doesn't have B carbohydrates on the cells. A person who's A doesn't have B carbohydrates on the cells, right? So no clumps. But either a B or AB person, that anti-B antibody is going to be clumping the cells. Similarly with B, right? O, no problem, no carbohydrates. A, well, the anti-A is going to go after that. Okay with B, not okay with AB, because again, there is the anti-A is going to attack the A carbohydrates. And this is the weird thing about AB. Now, there are other elements to matching blood. You know, there's RH, there's all sorts of other factors. But just with regard to ABO type, a person who has AB blood, AB red cells, has neither anti-A or anti-B. So theoretically, they can receive anything. So we talk about O being the universal donor. Well, AB would be the universal recipient with regard to red cells. Okay, and here's O. So again, O can receive O because, okay, there's no carbohydrates, but because they have both anti-A and anti-B, they cannot receive these other blood types. So anybody can receive O, but O can only receive O. And here's an example of blood typing little wells. Okay, so matching compatible blood groups is obviously critical for blood transfusion. If a donor's blood cells have a carbohydrate that is foreign to the recipient, then the immune system is going to produce these blood proteins called antibodies that are going to bind to those carbohydrates. And that clumps the cells together and in severe cases can actually kill the recipient. So it's one of those things where you have to be mindful. All right, so pleiotropy. Pleiotropy is when a single gene influences several characters. And going back to sickle cell disease, that is an example of pleiotropy. It results in the abnormal hemoglobin proteins and causes that disc-shaped red blood cell to instead be that sickle shape. But because of their shape, they don't flow properly in the blood. They tend to build up in clogged blood vessels. And again, you know, you mess with the structure, you mess with the function. So there's a cascade of things that go with sickle cell disorder, unfortunately. That's what a sickled cell looks like. All right, so I'm passing over the themes there. All right, so pleiotropy. You're going to have one gene affecting multiple characters. Polygenic inheritance. Poly says what? Many. So polygenic inheritance is where two or more genes, right? Poly number of genes affect a single phenotypic character. So it's sort of the opposite of pleiotropy, right? One gene affecting several characters is pleiotropy. Polygenic inheritance is where multiple genes affect a single character. And you generally see this with characters that have gradations, right? Height in people is controlled by several genes that are inherited separately. So collectively, these genes are going to determine a person's height. So many diseases, diabetes, heart disease, cancer, display this polygenic inheritance. It isn't like you're looking for one gene to contend with. And the neat thing is that polygenic inheritance is, you know, kind of an interesting example of interactions, that many smaller parts interacting can result in new properties. Now, brace yourself. You've seen the monohybrid cross. You've seen the dihybrid cross. Now we see what? Trihybrid cross. Right? So instead of 1 out of 4, 1 out of 16, it's, you know, 1 out of 64 is your, you know, lowest probability. So instead of 1 to 3 or 9, 3, 3, 1, you've got 1, 6, 15, 20, 15, 6, 1. You do not have to memorize that. But I do want you to see that, okay, from the two extremes, right, this would be heterozygous for both of these, but the possible combinations beyond that, right, because you've got 3 different alleles that are going to come in dominant or recessive. So when you're trying to calculate that, each one of these matings is really going to be, you know, talking about, you know, 1 in 18 and 8, you know, 8 times 8 is what? 64. So instead of 1 out of 2, being a number out of 4, or the 9331 being, n out of, 8, 4, 4 fours is 16. So you have the 4 possibilities and 16 possible offspring. Well, now we got 8 times 8, 64 possible offspring. And so, yeah, this would be an absolute beast to draw in an exam, and you will not be doing that. But I wanted you to see this. And the fact that if you think about most of the Mendelian stuff, it's one or the other, right? White or red. But here, because you've got multiple alleles, you wind up with a gradation, right? People don't come in just short or tall, right? You have the extreme of very short. You have what's sort of the average, right? So if you go in the middle here, you'd have, most of the stuff resulting in a generally average height, right? So the 15, 20, 15 crowd. And then all the way down to the other extreme of very tall. So when you see a gradation like that, it's often a polygenic thing. Human skin color is similar to that too. Right, so again, 3 alleles, independent assortment, boom. So yeah, that would be a lot to try and draw out in a test, so don't worry about that. Okay, epigenetics. Hopefully by now you've seen the video, right? And talking about this whole thing of this level beyond simply what genes do you have, right? And we talk about phenotypic characters resulting from a combination of heredity and environment, right? So whether human characters are more influenced by genes or the environment, the old phrase is nature versus nurture, right? Is there a characteristic that is more from the genes or more from, where that individual lives? And boy, this is one you can get into a lot of really good fights over. So really strongly contested. And the interesting thing is if you say identical twins, right? So these are people who have the same genotype. Right? Identical twins is one fertilized egg splits into 2, and you wind up with two fetuses. So these people have the same genotype starting out. But if you spend time with folks who are identical twins, you'll realize that the environment, things like, effects of personal choices are going to affect a person's traits. So in general, only genetic influences are inherited, and any effects in the environment are not usually passed to the next generation. But again, you start looking at things like epigenetics, then what? You had things like stressors that were causing methylation issues with genes. And so again, remember the business of methylation where the gene is bound, it can't be expressed, all that stuff. So in general, we only talk about the genetic influences being inherited, but now we're realizing with epigenetics that, wow, the environment can actually affect how genes are going to work in the next generation. So it's adding a whole level of interesting to this. This is an example of, a pair of twins who, made choices like different hairstyle, different color. So, even identical twins can look different because, again, this would be nurture, right? This would be, what's going on with that individual's choices. But here's another good one. There's leaves where, depending on the position of a leaf, so this is one tree, right? So all the leaves of the same genes because they're coming off the same tree. But depending on whether the leaf is more exposed to sunlight or less exposed to sunlight can influence what form that leaf takes. So it's kind of interesting that would be a great example of how environmental influences can affect things. All right. So again, now we're starting to realize this business of epigenetic inheritance, the transmission of traits by mechanisms not directly involving the DNA sequence. And again, most of us grew up with this whole business of, okay, here's your genes, here's what you get. But now they're realizing that components of chromosomes can be chemically modified by adding or removing chemical groups on the DNA and or those proteins, remember the histone proteins, protein components of chromosomes. So if you have something that keeps that remember you had to have the DNA unwind to get copied, to be transcribed into the RNA and on from that. Well, if it's basically tied up by chemical groups that are sitting on that DNA, it can't unwind. So what happens? That gene can't be expressed. So epigenetic modifications and then the changes in gene activity that come from that can be carried on to the next generation. But what is really interesting, and again, if you guys saw the videos already, you already heard some of this. Unlike alterations to the DNA sequence, chemical changes to the chromosomes can be reversed. Right, so things like dietary changes um, can actually reverse some of these problems. Okay, chromosomal theory of inheritance. This shows that genes are located at specific positions, remember the loci on chromosomes, and the behavior of chromosomes during meiosis and fertilization accounts for inheritance patterns. Okay, so going back to meiosis, right? It's chromosomes that undergo segregation and independent assortment rather. So we saw this looking at meiosis, and that explains some of Mendel's laws, because again, Mendel didn't know DNA, didn't know genes. He just knew these heritable factors. The next diagram kind of ties together the business of meiosis and looking at in terms of a dihybrid cross. Right, so kind of big and hairy. So I'm going to do is I'm going to go to a zoom in on the next slide. All right, so here's a parental generation. We're looking at dihybrid cross. All right, purebred for the recessive, purebred for the dominant in the parents. All right, so by meiosis, again, because they're homozygous, there's only one possible gamete they can produce. And so by definition, the F1 generation are all hets, right? They're all heterozygous. Now we're going to do is cross our F1 generation, right? So these guys are starting with this. And through meiosis, you've got two possibilities, right? One where the alleles from one parent travel together. Let me put it differently, sorry. Where the dominant alleles travel together or the recessive alleles travel together. But equally probable is that they arrange differently, right? So again, they assort differently from each other. They don't have to travel together. So if you look at this, these are two equally likely ways these see metaphase 1 take place. Well, then if you go to metaphase 2, okay, because the dominants did remain on one side, the recessives on another, right? Only one, type of, chromosome is going to wind up in each gamete. It's only going to be the dominants. It's only going to be the recessives. And then say, okay, so once this divides up, there's your gametes, right? So 1/4, 2 copies of the dominant. 1/4 is 2 copies of the recessive. But again, equally probable is you had this happen in metaphase. So now we look at these gametes and 1 out of 4 that you have recessive one, dominant of the other. Similarly, dominant of one, recessive of the other, right? These are equally probable events. So 1 out of 4, you get these possible gametes, right? So when you're doing these crosses, remember that dihybrid cross diagram, fertilization, and again, this is if you made a bunch of these flowers. It's not just like grab one flower and count up, one set of peas. You generally do this with dozens and dozens of individuals to look for their actual patterns. Anyway, fertilization among the F1s, because you have all these possible gametes, wind up with that 9:3:3:1, right? So I talked about that 9:3:3:1 phenotypic ratio in the F2 generation. Do not worry about genotypic ratio in a dihybrid cross. That would take a while, so don't worry about that. Okay, I'm just going to skip over here. Linked genes. Okay, I think in the review notes I put down that, not to get crazy about this, the point of understanding linked genes is that genes that are near each other on the same chromosome tend to travel together. Again, go back to that crossing over, right? We had the strands overlapping each other and coming away with pieces of each other. If you've got two genes that are right next door to each other, the loci are very close together, the odds of them getting split apart from each other is relatively low. And so we tend to see linked genes that are located near each other on the same chromosome, they tend to travel together because the odds of them getting divided are relatively low. So they're often inherited as a set and therefore kind of blow Mendel's law of independent assortment out of the water. Now, so far, and again, that's as far as you have to know, nothing beyond that. Um, Now, we've been talking about autosomes all this time, right? So, patterns of genetic inheritance involving genes located on the autosomes. So, now let's look at the sex chromosomes. And I've got to do is take a pause here, and I'll return to finish this in a moment.